Okay, so therapeutic strategies. You have heard a lot today from different people. They presented examples of their work or work they've heard about and meetings and papers, etc., how to challenge this condition. So we can think about, I took this figure from a recent paper by Dr. Puccio, whose work was mentioned a lot today. And so she summarized basically the therapeutic strategies in 5 broad categories. The first being the attempt to increase frataxin expression. And we heard a little bit today about agents, even natural agents, such as PGC1 alpha, for example, which is a controller of mitochondrial biogenesis and naturally should increase frataxin. And so pioglitazone is an example of a clinical trial that is ongoing in Europe, which does exactly that. There is also, and we heard a lot about this from Jim, Dr. Rushi, how to try to target specifically the expression of frataxin. For example, through the use of HDAC inhibitors, the category of drugs which facilitates the expression, the synthesis of the mRNA of the protein. Something that we have not talked too much about today is the idea of replacing the protein itself. So while this approach addresses the DNA problem, so the problem of the DNA being transcribed into mRNA, which then gets transcribed into protein, another possibility is to go and replace the protein. And so there have been attempts to deliver the protein directly to mitochondria, directly to cells, using a biological trick, adding basically a little protein to the frataxin itself. And this protein derives from a virus, which is able to penetrate cells, as you know. And so this would be sort of a chaperone that would bring the protein inside the cells. Of course, there are lots of technical issues to be addressed, but Dr. Payne, Indiana University, has pioneered this. He has done it in cell culture models, the neurons I talked to you about earlier, and he's done it also in the mouse model, Dr. Puccio's mouse model, with some success. And so hopefully this approach will mature in the next future. And then, of course, we heard a lot about gene therapy. So this is, in a way, I guess it's a hybrid between replacing and fixing the DNA and replacing the protein. It's a way to replace the protein by supplementing a new set of genes, basically. And we heard about iron chelation from Rob Wilson. He mentioned that some iron chelators, so the idea, I can remind you briefly, is to remove the excess iron from mitochondria and put it back in the cytoplasm where it belongs, and to avoid the cells sensing a starvation for iron and actually worsening the situation. New molecules model after this one, which may be more efficient, as we heard today, weak chelators that can do exactly that job. And then we heard a lot about these mitochondrial drugs. And so these are drugs, for example, idebenone, some other antioxidant. Well, idebenone is really an analog of Coenzyme Q. I'm sure you've heard of Coenzyme Q over and over, everywhere. Idebenone and some other analogs, A0001, for example, that David Lynch talked about before, and new brothers and sister compounds. So what are these drugs really doing? And I think this is the only aspect of this. All the other categories that I just mentioned are pretty self-explanatory. You replace the gene, you try to replace the protein, et cetera, et cetera. But this one is, it's always been a little confusing to me. So we always call them, as you see here, antioxidants, mitochondrial activators. It's very vague. And the main reason for that is because despite these drugs being the ones that have been tested the longest, I think, in this disease and in other mitochondrial diseases, are probably the ones whose principle of action we understand the least. This is my personal opinion. I'm not speaking for anybody else. But I just want to bring you back very quickly to... Don't worry, I'm not going to give this presentation again. But just this slide. Okay, so I mentioned to you this, that these electrons that come from these nutrients, they have to be transported by this chain of enzymes. And a very important factor is coenzyme Q. I didn't mention this before. I skipped on it because I knew I was coming back to it now. So coenzyme Q is a fundamental cofactor that shuttles electrons between these enzymes. And all these drugs that we've been talking about, edebenone, coenzyme Q10, of course, A0001, possibly, are very similar to this natural component of the respiratory chain. And I think at this point what we would like to understand, or I would like to understand personally, is whether these drugs are meant to substitute this. So by substituting to this, facilitate in some way the transferring these electrons at sites, at places where because of the frataxin deficiency and the iron cluster assembly deficiency, the electrons cannot pass very well. And so it's like we're introducing new electron donors, we call them, which could take electrons from here, for example, and bring them straight downstream of the blockage. That's one idea. The other simpler idea is that because this molecule is also an antioxidant, and a quite potent one, in fact, if we're not just, I wouldn't say just supplementing, but mainly supplementing an extra source of free radical scavenging, so eliminating this potential damage from a reactive oxygen species. And I think this question is still open, and I think it's upon the scientists who designed this molecule. A great scientist was mentioned today, great in the sense that he's designed great molecules for cancer therapy in the past, and is now put his head to designing this molecule, but also to understanding how they work, and Sid Heck at the University of Arizona. So now, oh no, I have to do all this, quick. The pipeline. So this is the list, basically. You have seen this before. Yeah, because I modified it a tad, just a little bit. Well, I raised the green bar here, for example, to preclinical. I took the liberty of putting... So I don't know, this idea of what is preclinical is also a little bit of a vague concept. It includes everything that happens in the lab between having an idea and actually going to a human being. Everything else is preclinical in a way. So, but as you can see here, the gray bars are all those compounds that in some way resemble coenzyme Q. Look how many we have. So for years, I think this has been the main focus, not because we thought it was the only focus, but because it's what was available at the time. I mean, the first few years of the pipeline. And some of these have been brought all the way up to phase 3, as you know, edebenone, clinical trial, and many others are under phase 1, phase 2 clinical trials. And we heard quite a bit about this one, so I'm not going to reiterate all that. I just want to briefly add a few notes about the ones that have not been discussed so much. So EPO, I know you heard about this. So, but how is it supposed to function? And that's something that we're also trying to understand, despite the fact that EPO has reached phase 2 clinical trial, I think in Italy and also here? No. In the States? Yes? Italy, right? That's what I thought. And again, there was a mixed bag. I think there was some evidence that EPO raises the levels of frataxin. The good thing about EPO is that it's a natural hormone, so your body makes it. The bad thing about EPO that Rob Wilson mentioned before is that it might increase the amount of red blood cells too much and make your blood really thick, which is not a good thing for the heart. So what we're exploring now together in conjunction with small biotech, basically, is to see whether so-called hypomimetics can also work to increase frataxin. And that's important because the hypomimetics may be able to act through the same mechanism so the same mechanism of increasing frataxin without actually activating the hematopoietic response. So you would have the benefit of making more frataxin without having the drawback of having too many red blood cells. But this is still very early on, but we think it's an interesting perspective. And so we talked about HDACs, definitely. We talked about interferon gamma. We didn't talk very much about this. So this is well, it's an old drug. It's not a new drug. I mean, it's not even a drug. Again, it's a natural substance that your body makes. It's involved in immunity, inflammation. And serendipitously, an investigator in Italy, Dr. Testi, found out that interferon gamma increases frataxin levels. Again, the mechanisms are not clear, and they're studied, actively studied. FARA, I just want to mention that FARA, one way or another, is involved in all of these initiatives. Sometimes completely, sometimes partially, but we're involved in this both financially and intellectually. So we do consult with all of these scientists. We try to advise, and we certainly provide a lot of help. David and Jennifer and everybody, and you guys, obviously, with eventually, when it gets to the point to testing on biological samples or even in people, some of these approaches. So I was going to say that interferon gamma increases frataxin levels. That's promising because interferon gamma has been studied extensively in humans. So that if that worked out, that would be one of those cases where you can sort of piggyback on an existing drug and repurpose it. So it was used for, say, treating multiple sclerosis, but hey, if it can be used for frataxin, at least we know everything about the safety issues of this drug because it's been used for years in humans. And there is not the only example of things like this. So gene therapy, I think these are two examples. They're still, as you can see, the bar is low. We're still at the early phases, but we're extremely excited about these ones. You saw how promising the results are in the mice. So it's certainly something we want to keep pushing for. That frataxin I mentioned before in the previous slide is that trick to replace the protein in the mitochondria. This may become, as I said before, may expand in the near future. And then in red here, we have things that actually don't exist yet. So these are multiple different attempts to identify new drugs or repurpose old drugs through pharmacological screenings. And what the red bars really represent are the platforms on which to screen the drug. So, you've heard before, there are libraries, of hundreds of thousands of drugs. The drugs are there. In many cases, the issue, as David mentioned many times today, is to figure out what are the good endpoint. What is telling you exactly in a super simplified manner, something that you can appreciate very quickly looking at a cell culture dish, that this drug is benefiting the cells which don't have enough frataxin. And so that's the trick. I mean, I think, is very important to develop reliable and reproducible assays just to start screening drugs. And so these are three examples of, I'm not specifying here what they're exactly doing, and I don't want to bore you too much with the details. But there is a lot of effort going into developing appropriate screening tools for drugs. And so I think I don't really have much to add at this point, and I guess if you have some more questions, Jennifer, Dave, coming here. Thank you.